Namaste. So we are going to talk about the cell inclusion bodies which are usually found in the prokaryotic organism. The first one is the carbon storage polymer, which is found as the PHA in the prokaryotic organism. The PHA basically stands for the polybeta hydroxy butyric acid, which is a type of lipid which is formed from the beta hydroxyl butyric acid units. So number of these units are joined together by the ester linkages and they form together the PHA. So the monomer for PHA is hydroxybutyrate. Depending upon the individual organism, the PHA can vary in length from like the minimum number is 3 and up to 18. It can vary upon the individual. So what is the function for this PHA? So PHA basically stores the carbon products which is present around the organism in its environment. So when the organism has like these carbon product in the environment, it is going to take those products inside and then it is going to store them. And when the conditions are as such or when the conditions are appropriate, it is going to break those stored carbon sources down and then it is going to use those as its energy sources. Similarly, there are other minerals which are being stored by these prokaryotic organisms. The second one is the phosphate. So phosphate is present as inorganic phosphate in the environment, which is taken up by the prokaryotic cell, and then the cell is going to store it as a granular structure, which is known as the polyphosphate. And as per the requirement, the cell is going to degrade those stored phosphate material and going to utilize the phosphate, right? It can be in the DNA or for the bilayer. It is to note that the phosphate is often a very limiting nutrient in the prokaryotic organisms. Hence, when there is abundant of phosphate in the environment, it is very essential for the organism to store the phosphate in its cell. Moving on, we have the sulfur. So the sulfur, it is present as hydrogen sulfide in the environment. So for example, we have the example here, isopropyl butyrate, that is an example of a sulfur bacteria which is storing the sulfur in its inclusion bodies, right? So what happens is hydrogen sulfide is present in the environment. It is taken up by the bacteria, the sulfur bacteria. So when it enters the sulfur bacteria, it undergoes oxidation. Why it undergoes oxidation? The cell, the bacterial, it requires electrons, right? So it can require electron either for chemolithotrophy or autotrophy, whatever the process is. Regardless of the process, the electron is taken out and an elemental sulfur is formed inside the cell and it is stored as elemental sulfur. So we often have observed that when the bacteria has abundant of H2S in its environment, it stores the sulfur inside as the elemental sulfur. And when this elemental sulfur is stored inside the cell, it appears as if there is granular structures in the cytoplasm, right? But the trick here is that the surprising fact here is that the organism is not actually storing these granular structures in the cytoplasm, but rather it is present in the periplasm. So when the H2S is being oxidized into elemental sulfur, it expands the periplasm and hence we see these structures in the periplasm, not in the cytoplasm. Then as the concentration of the H2S, it decreases in the environment, we see a change. The elemental sulfur then again goes through the oxidation and it is converted into sulfate. And as the sulfate is being produced, its concentration increases, we observe that these granular structure also start diminishing, and we see a plain structure as compared to this. So the periplasm, again, when the oxidation between elemental sulfur and sulfate is going on, the periplasm, it will contract. So it will contract inwards, right? And when the H2S was being converted into elemental sulfur, the periplasm was being contracted outwards and it was swelling. It was the periplasm where the granules for the elemental sulfur were being stored. Hence, we see these granular structures. The last one is the carbonate minerals. So, usually these are seen in the cyanobacteria. Often, what happens is in the cyanobacteria, the carbonate minerals are found on the outside of the, like they are attached on the outside of the cell of the cyanobacteria. But in some cases, it can be intracellular. That means it can be present inside the cell. So we have one example, that is the Julio Margarita, in which the granules are present inside the cell. So the carbonate mineral here, which is stored, is called as the bentonite, which is made up of the barium, strontium, and magnesium. So, why is this important? This is important because here comes the concept of biomineralization. What do we mean by biomineralization? It is basically the microbiological process of forming minerals. So, what happens is, as the cyanobacteria is depositing these carbon minerals inside, it is going to form deposits within the site where it is present, in the environment where it is present. Hence, there are mineral formation that is being taking place in that particular environment, and hence minerals are being formed because of microorganisms. So simply it is biomineralization. Also, it is not known why these cyanobacteria are able to, or why are they doing this biomineralization, but it can be that it is to maintain themselves in that particular environment. Thank you so much for watching. I think that's all that I have to say. Thank you. Bye bye.